Assistant Professor in BB College, Asansol, West Bengal, India. Today we are going to study a very popular topic called gene regulation in prokaryotes. Gene regulation is a topic that deals with those genes that are under the control of certain regulatory elements. Not all genes are regulated. There are two types of genes, in fact. One is called the housekeeping genes. Housekeeping genes are those genes that are involved in vital metabolism of the cell, like the glycolytic pathway, Krebs cycle, or the lipid metabolism, amino acid biosynthesis, all that. These enzymes that are the respective genes of those enzymes should be always expressed in the cell. If any one of them is blocked or regulated, the function of the cell will be inhibited and the cell might die because of that. So these genes are called housekeeping genes, or they are also called constitutively expressed genes rather. But there are other set of genes that are under the control of environmental factor, and those are called regulatory genes. These genes will only be expressed under certain environmental conditions. For example, in case of bacteria, say Lac Operon, that constitutes of 3 genes. This operon, in other words, the structural 3 genes that are under the control of these operons, are only switched on when the environment, that is the media, contains lactose. When lactose is given, then only the enzymes needed to break it into galactose and glucose has to be synthesized. That enzyme is called beta galactosidase. So this enzyme is only synthesized when lactose is present. In its absence, beta galactosidase enzyme is not synthesized, and it is under the control of this Lac Operon. So Lac Operon is a regulatory operon, or regulatory gene, one might say. So there are genes that are controlled. If you talk about temperature, there are heat shock proteins that are expressed only during when a cell is subjected to high temperature or low temperature, and so on and so forth. So basically, these regulatory elements, the regulatory genes, are under the control of environmental factors. So we can get into some example of this regulatory gene. Say in case of prokaryotes, genes are present in groups, in clusters. Several structural genes. For example, we are talking about Lac Operon. Lac Operon is a regulatory operon. It consists of 3 genes, structural genes: Lac Z, Lac Y, and Lac A. Each of them expressing a different protein. Lac Z, it forms a beta galactosidase enzyme. Beta galactosidase. Lac Y, it forms Lac permease. Lac permease is an enzyme that moves to the cell membrane. It is a cell membrane enzyme that allows the transport of lactose in the cell. Lactose will enter in the cell because of this Lac permease. On the other hand, there is another enzyme called transacetylase. This enzyme is also grouped along with Z and Y, but the function of transacetylase is no way related with the Lac metabolism. These three genes are under the control of a single regulatory element that has been divided into three parts: the inducer element, the promoter element, and the operator element. To remember, IPO in order, Indian postal order, might be kept in mind. So these are the structural genes, and this is regulatory elements or genes, whatever you say, and this together, the whole structure from here to here is Lac Operon. This is present inside a bacterial genome. This is a bacteria, this is a bacterial genome, say. A certain part of it is basically this Lac Operon gene, the operon that constitutes that part. Now, let us see how this operon is regulated. Let me wipe it out completely. So, there are two conditions by which Lac Operon could be understood. One is in absence of lactose, another in presence of lactose. What will be the behavior of a bacterial cell like E. coli growing in a media in which lactose is absent and in a condition when lactose is present? In absence of lactose, what will happen? The I gene, it produces a transcript that is encoded into a protein called Lac repressor. In fact, there are 4 proteins of same molecular weight, so it is a homotetramer. This is Lac repressor. This Lac repressor, in absence of lactose, it goes and it binds to this position, promoter operator region, and thereby it blocks the waiting RNA polymerase, you can say, this enzyme RNA polymerase. This had been waiting to bind here to start transcription, but since it is already been occupied by a protein called Lac repressor, this protein, and that is why it cannot bind, the RNA polymerase cannot bind here, and transcription cannot take place. So the gene is said to be switched off. This is how a gene remains, the operon, the ZYO, in fact, these 3 genes together, they remain unexpressed in absence of the environmental factor here, that is the carbohydrate lactose. So now the next situation, when you add lactose, then what happens? Let me wipe out this part only, because the rest of the areas will be needed. So this was the structure of a gene, operon rather, Z, Y, and A, I, P, and O. So normally, in absence of lactose, I was synthesized into a Lac repressor. When lactose is being added, it is immediately converted into a product called allolactose. Allolactose. The enzyme involved here is same, that is beta galactosidase. because the basic level of beta galactosidase is already present in the cell, and there is enough to convert lactose to allolactose, a particular type of metabolite that binds with this Lac repressor and changes its conformation so that it is not able to bind with the, say it changes its conformation in this way and makes it inactive. When this Lac repressor is inactive, it cannot bind to operator region, promoter operator region, and the RNA polymerase that had been waiting here now is able to associate with this part and start transcription from here. A polycistronic mRNA is synthesized. From this polycistronic mRNA, the three different enzymes already been told that are made. So genes become switched on, but this is not the positive control. The positive control of Lac Operon only begins when two other factors called catabolite activator protein and CAMP. This complex catabolite activator protein CAP and CAMP, until they associate to this part of the genome, the RNA polymerase is not able to transcribe at 100% level. In the absence of these two, the transcription level is merely 2% of the total. But when CAP-CAM bind there, CAP-CAM means the catabolite activator protein and the CAMP protein, when they bind, the transcription level now reaches to 100% maximum, and this is called positive control. The gene is said to be switched on now. So that is how the gene of Lac Operon is regulated. Now let us see what will happen if you add glucose to the media. In presence of glucose, what will be the behavior of Lac Operon? Let me wipe it out again. In presence of glucose, the bacteria will show a kind of graph, a kind of growth that is being called dioxic growth. It will first utilize glucose. So it will show a lag phase, then a log phase for glucose. When glucose will be completely consumed, then only lactose will be utilized, and that will show another lag phase and another log phase, followed by a stationary phase and then death phase. So this is called dioxy or dioxic growth pattern. This is time, and on this side you can say absorption at A600 nanometer. So this is the level. At this position, the bacteria, when the glucose is completely consumed, it starts producing beta galactosidase enzyme, and it metabolizes this is for beta galactosidase, in brief beta gal I would be saying. So in presence of glucose, this kind of growth pattern, when glucose is present, lactose cannot be utilized, and this is called type of glucose effect or catabolite repression. How glucose execute this effect? In presence of glucose, the CAMP level, when glucose is present in the medium, the CAMP level that is necessary to bind with the CAP, forming CAP-CAMP complex, is not being formed. The CAMP level is very, very down. And that is why the CAP-CAM complex could not be formed, and the Lac Operon cannot be switched on to 100% level. So how glucose reduces the concentration of CAMP? There are few reasons for that. When glucose is transported inside the cell through the enzyme called E2A, earlier it has been called E3. When it is transported through this E2A, it is immediately phosphorylated to glucose 1 phosphate, a charged molecule, so that it cannot be allowed to move away from the cell. That is why the glucose is immediately phosphorylated to convert it into a charged molecule. The phosphate it is given is from this molecule E2A enzyme. This enzyme is initially phosphorylated, and that donates its phosphate to the glucose, and now it becomes dephosphorylated. Now the phosphate that E2A gets is through the group translocation process, and that is a long process. It starts in the cytosol from the pyruvic acid, rather from the PEP, from the PEP to pyruvate or pyruvic acid. The PEP transfers its phosphate to E1 first. E1 gets phosphorylated, E1P. E1 donates its phosphate to another protein called HPR, heat shock protein. HPR now gets phosphorylated, HPRP. HPRP donates its phosphate to E2, and E2 in turn transfers that to glucose, incoming glucose, to convert into glucose for 1 phosphate. In the meantime, E2A, it becomes dephosphorylated. A dephosphorylated E2A, or E3 you say, the earlier one, does 3 things. It inhibits the adenyl cyclase enzyme present in the membrane. This is the adenyl cyclase enzyme, the enzyme that converts ATP to CAMP. This is being blocked. The Lac permease is being blocked, so lactose cannot enter now, and the transporter for CAMP is opened. So whatever CAMP is present in the cell is expelled out of this cell, and that is how the level of CAMP decreases in the cell, and that cannot bind with the cap because it is less in the cell until glucose is completely utilized, completely consumed. CAMP level remains down because of these reasons. Then when glucose is completely consumed, CAMP level increases, CAMP complex is again formed, and Lac Operon is switched on, and lactose is now metabolized. So this is all about Lac Operon for today. Thank you very much.